able to see the
can
projection?
continue. Yes,
Okay.
just
Yeah,
continue.
so the topic is there are three concepts that we are going to cover this morning: pathogenicity, virulence, and infection. So there are words individually, and it depends on the context, and you will get the meaning when you get the concept. So I don't know my projection. So we're going to try and analyze each one, but remember pathogenicity and virulence, they share some factors in common, and sometimes they're used interchangeably, and even in infection, sometimes it is used to mean disease, sometimes it is even interchanged with virulence and all those. But one needs to get the concept of what's being talked about, get our reading at that moment in time. So they refer pathogenicity, virulence, and infection. They refer to some aspects like between bacteria and humans during infection. So you know, bacteria exist on their own, humans exist on their own. Sometimes they come together. And that coming together is mostly the bacteria coming into the human being. And that is what is broadly called infection. But there are many other things that place when we come together, and that is the interaction. They interact, they just don't come there and sit and nothing happens. So when they interact, this interaction is also referred to as so there are fluctuations. Have your phone nearby. When you lose me, you call so that I know when to stop. It's saying the internet is unstable. So yeah, we were talking about the interaction, and it is also called host-parasite relationship. So it is a relationship between the host, who is the human being, and the parasite, which is the bacteria. And in that relationship, there are some alterations that occur in some physiological and biological mechanisms, and involve both the infected human, and for them, some of the areas which are affected are the immunological mechanisms, and these ones will be dealt with under the subject of immunology. It's not the changes, immunological changes that occur in the human being due to that bacterium that has got into him. And some of these may manifest, may develop into disease. There might be some manifestation, to talk about signs and symptoms which constitute manifestation. They can occur because of that interaction, and many factors that come into play before disease can develop. So some of them might not develop into disease, and some of them will develop in the human being. And then there are also changes which occur in the bacteria. And they will vary from one bacteria to another. Basically, the bacterium is getting into a human body, which has a lot of physiological and other mechanisms that will sustain it. So we were talking about the infecting organism. It also undergoes some changes because of its presence in a new environment that is not so, wow, is this going to work? So we start with the first one, if I can be heard. That is pathogenesis. You can hear. Can you hear?
You're breaking.
Okay, the first one is pathogenicity. I don't know what we can do to make this work. So I have to see what did I have on the phone. I'm breaking, but you can hear it.
Yeah,
Can you hear it?
your words don't get finished though.
Hello?
Doc,
Can you see the screen?
we can see the screen. The problem is your voice.
They are being cut.
Yes.
Yes?
Yes.
Okay. So just to save me time, can you also send this link to the phone? Up. We can use phone audio. Although it's going to bring a lot of echo, I can leave this and use the phone audio. So on with what we have. So the pathogenicity, if one looks at the word, it is a terminology that refers to the ability or the potential of an organism to cause disease. So when one gets an organism in this context, the organism is a bacterium. Any bacterium, one gets it and one wonders whether it can cause disease. Yes, that ability or the potential of an organism to cause disease. So it is a characteristic which varies among the bacteria, whether at the genus level, that is among the genera, whether it's the species of a genus, or whether it's the same one species. There are various strains, and pathogenicity would still vary among those strains. You can get one or two organisms of the same species, to get one which has potential to cause disease and one that doesn't. It's something, a characteristic that varies. But remember, it's just that ability or potential of an organism to cause disease. So it may be there, it might not be there. So based on that characteristic, bacteria are described non-pathogenic, which is where they don't have that ability or they lack that potential to cause disease. And then secondly, you have pathogenic bacteria, those which are really known to be associated with disease causation. And then you have opportunistic pathogens. This particular one could be in A or in B. They could be organisms which are regularly known as non-pathogenic. They are described as what are we going to do with this? It seems like we are facing challenges. I don't know why I don't have audio. Let me get it. and get audio on my phone. Just in it, I fidget with my phone, see if I can get my audio. They are not different organisms from the ones you have in A and B. They are causing disease. Can you still hear? Can you still hear me?
Yes, Doc.
Can you hear? Okay. So,
Yes, we can
the
hear.
phone has gone off also. So we will just go now this way. Okay, now the non-pathogenic bacteria and the pathogenic bacteria, they can cause disease in a person because of the altered immunological defense mechanisms in that person. And this alteration lowers the immune defense mechanism, so weaken. So some organisms which are known as non-pathogens or regular basis end up causing disease in a person who is immunocompromised and that is an opportunistic pathogen. At that moment in time, it's labeled an opportunistic pathogen. So the ones which are known as regular pathogens, pathogenic bacteria, they can still cause disease. A person is otherwise healthy, but when they cause disease in an immunocompromised person because of lower immune defense mechanism, it requires the label opportunistic pathogen, or the result is of an opportunistic infection. So from the word pathogenicity, there's the word pathogenesis, which is where there is what do you suggest we do here? Because this now is difficult.
Are you using Wi-Fi or data?
I'm using my, which would eventually end up with loss of and gets into the new host. And what goes on, the changes that occur in the host and the organism, all those would constitute the process of pathogenesis. So it includes manifestations associated with the. So these mechanisms involve the infected person and
Yes, Doc, we can still hear you.
the person, and we have already brought up, but it's also under pathogenesis. Various bacteria and different process characteristics or properties which increase the density. These properties increase the potential of their ability to cause disease. Such property is transmissibility of An organism easily passed on from one place to another. That is transmission. Passed on from person who is infected to another, that is communicable. There are some organisms which are not easily transmitted and they are not easily passed on from an infected person. So for those, their pathogenicity will be low, but there are some which are easily can be transmitted from an infected, and that exists. How or decrease cost. So transmission from one host or reservoir to a fresh host. And characterize ability to share in large numbers in body fluids or secretions. So if there are many organisms at the same time a person is being exposed to, they are easily transmissible, or then the chances of organism passed on to another person are higher than if there are fewer. The larger the number, the better. Okay, remove your notes. You send your notes to your colleagues, and I wanted to read mine. So how do I and continue? Zoom. So no, we have not reached there. So we are talking about the properties which increase pathogenicity of some bacteria that possess them. There's another characteristic of being able to survive under unfavorable conditions, like outside the human host. There are some organisms which survive better, others don't. And then there are some organisms which develop what we call bacterial spores that enable them to survive in the environment. No matter how harsh the conditions are, they are able to survive there. Then another property that is important in pathogenicity is infectivity. This involves the organism's ability to attach itself on tissues. And then after touching itself, it should be able to survive wherever it has attached itself and continue by penetrating the first line of defenses at that site. So the first line of defenses would be the skin and mucous membranes, and that is where the organism gets to first, attaches itself, and if it's able to survive, then it will continue to break through the skin and mucous membranes. So some organisms are able to do that better than others, and those that possess it, that ability, they do better. Their potential to cause disease is higher than those that are not able to do it. So that infectivity, it is more difficult in some parts with intact barriers than the ones where the barriers are broken. So it is easier where the surface is broken. It could be a broken part of the skin. It could be a broken part of the mucous membrane. So you'll be hearing a lot about organisms that infect human beings through broken skin or bruises, no matter how shallow, no matter how small a tear is on the skin of mucous membranes, they find their way in because it has been weakened for them and they are able to do that better. So infectivity is also influenced by a number of factors. The number of the infecting bacterial cells, the higher the chances of some of them succeeding in attaching themselves and going through. Victor, what are you saying? Victor, are you talking? Then the phase of growth. When you did the bacterial growth, the physiology, There are about 4 phases, and there is one phase where the bacteria multiply rapidly, and the metabolism is high, and they are very aggressive, and that is the exponential phase or logarithmic phase. If a site in the body is exposed to bacteria in that logarithmic phase or exponential phase, the chances of them succeeding in infecting that person are higher than a person or a site that is exposed to bacteria in the lag phase or phase of decline. Those particular ones are not so aggressive. So the phase of growth is also important. Then properties of bacteria. These bacteria that have the potential to cause disease also possess some properties which enable them to harm or destroy tissues. So we are going to talk about those in a short while. They use those, and those would enhance their infectivity as well as harm human tissues, part of the disease process during infection. So a person can be exposed to an organism that is able to attach itself and that is able to penetrate, and there are possible results. It doesn't always result in disease. One, there can be resolution. The process can be arrested somewhere before the disease develops. So that is resolution of the infectious process. And then secondly, and more importantly, is obvious illness resulting from that interaction due to the organism. This can take several ways depending on the particular organism. It can occur as a localized lesion at the site of entry due to associated inflammatory response. That is commonly seen at a site which is infected by some bacteria that are associated with pus production. And pus comes about as a result of the inflammatory response or defense mechanisms coming into play to try and get rid of this organism. So the presence of that organism and the inflammatory process mingle, they work together to produce this obvious illness. And then it can also take a form of a systemic illness, that is an illness that involves a specific organ or tissue within the body, inside, in the inner part of the body or in the systems. Or it can be a disease that is spread, widespread within the body, involving multiple parts. Another possibility is development of latent or asymptomatic or subclinical infection. This can progress later into a symptomatic illness, so it is also important. And then there are some infections which go unrecognized. They are signs and their symptoms, but they might resemble others or caused by other organisms, or they might be too mild and they are overlooked. And then lastly, there can be a chronic carriage of an organism. There are some organisms that are associated with chronic carriage, and the person develops a chronic carrier state or status. So as you cover the various organisms, you encounter some of these conditions as a result of that interaction between the bacterium and the human body. So the chronic carriage of pathogenic organism, this is important. It doesn't occur all the time. There are some specific pathogens which are associated with it, and even with them, it doesn't always occur. So there are some organisms where they infect the person stands a chance of developing a chronic carrier state. But it doesn't happen always, but the chances are higher than with other organisms. And when it develops, it's characterized by a small number of viable organisms. They reside somewhere within that person, a small number, maybe dividing very, very slowly, and no clinical manifestations at that moment in time. So this can develop after the clinical illness that we mentioned, whether it is systemic or widespread or involving localized. So they can undergo recovery clinically, maybe even after treatment, and look like they have recovered, but somewhere within them, they are harboring these organisms in small numbers without any normal manifestations being encountered. Chronic carriage can also develop after asymptomatic infection or unnoticed infection. But whenever it occurs, it is important because there's a possibility of those organisms being a source of infection, which leads to symptomatic illness. And this can still occur in the person, in the carrier, the person who is harboring them. It can, one would think of it as a flare-up or something. They would begin to multiply rapidly, and the person will develop manifestations, and this would be symptomatic illness now in the carrier. But they can also be transmitted. Small as the number is, there's also a chance of transmission to another person. When that happens, that particular person can develop as the illness itself. So that is an important significance of this organism. They can be a source of infection that will eventually become disease. Secondly, one can develop long-term complications associated with the illness caused by the organism. It might not manifest clearly, but complications might show. And there are some severe long-term complications which can arise in relation to this carrier or to the organisms that are being carried by that person. And the last point about this condition is that it is detectable by laboratory tests. So where it might be suspected, then there are some tests which can be done in the laboratory, whether bacteriological or immunological, that would detect the presence of this of the organisms in that person. So the second terminology, virulence, this is actually a component of pathogenicity. It is like pathogenicity, but it is a measurement. It's measured. The other one, you're just asking whether that organism has the ability or doesn't have. For this particular one, that ability is measured, and it is what is called virulence. So it is defined as the capacity of an organism to harm human tissues or to cause disease, or the degree of pathogenicity.
Binursi share.
Pardon? Somebody is saying what? What are you saying? Okay, so
just continue, madam.
You can hear me?
Yes, we can hear you.
Let's continue. Okay, so I was talking about virulence, and I'm saying sometimes it's actually used instead of pathogenicity. But as I said earlier, you have to get the context under which the words are being used. And I said virulence is a measurement of pathogenicity, degree of pathogenicity. For pathogenicity, you just want to know whether it has the ability or whether it doesn't have the potential. For virulence, you want to know how much potential. So it is the capacity of an organism to harm human tissues or to cause disease or the degree of pathogenicity. So it is a measurement of pathogenicity, and there are experiments that are performed to do this, to measure virulence, and it is measured in relation to host resistance. That is how you can know it's how much capacity, to harm those tissues, and it is basically useful in comparison, comparing the various organisms. Could be species. You want to know how much or the capacity or the degree of pathogenicity of the organisms within a species or within the genera or two different organisms. So it is a component of pathogenicity, except that it's measured to some extent. And the factors which influence pathogenicity are also important. They also influence virulence. And it also varies as pathogenicity. But an organism can be described based on virulence. And this could be a virulent organism when it has no capacity to harm human tissues or to cause disease, or its degree of pathogenicity is very low. Or it can be virulent when it is capable of harming tissues. Mostly, most organisms are just virulent. They can harm tissues, or they have a moderate degree of pathogenicity. And thirdly, an organism can be highly virulent. This is one that causes damage very fast, and disease manifestations are very severe. And the disease, if it is there, it progresses very fast and becomes worse within no time. Those are organisms which are described as highly virulent. So this characteristic virulence, it also has some factors that enable it, that would enable the organism to have a high capacity or to have some capacity to harm tissues. And these properties of bacteria which cause disease are referred to as bacterial virulence factors. They enable it to harm the tissues or to make it that virulent. So bacterial virulence factors are properties of the organism which enable it to harm or destroy tissues during infection. So these are basically the bacterial properties, but as we saw earlier, there are also changes in the human host that contribute to what would be seen as a manifestation of a disease eventually. But if we confine ourselves to the bacterial aspect, the virulence properties of bacteria are mainly determined by its genetic composition. That one will determine many things in the organism, the way, the structure, the method of metabolism, etc. And some of those would reflect also in the virulence. So it is mainly determined by genetic composition. There are also some genetic components that code for some of the factors that we are going to mention as virulence factors. But as I said, it codes for many things related to the organism as a whole that might also contribute in its virulence. Then biochemical properties, again, these two biochemical properties and structural components, they are at the end of the day determined by the genetic composition. But there are some specific biochemical properties that would harm tissues, and there are also some structural components which are useful to the organism in causing harm, and we are going to mention them. So the virulence properties are numerous, and they share some characteristics, the way they function. Some of them share this method of activity, and some have different mechanisms of action. So there's some shared mechanisms and there's some which are very specific. So the number and types possessed depend on the pathogen, on the bacterium that one is dealing with. So it is not consistent. It depends on that organism. And even within one organism, it might not be consistent. But there are some basic ones which are consistent. Now, each factor can be possessed by several different bacteria, and that is what translates into that shared mechanisms of action. They share some factors also, and then one or more factors can act in one disease process. So you can get many of these factors causing harm in different ways, and altogether that would amount to harmful effects and the manifestations of the disease. So to mention them, it's important to classify them, and there are many different methods of classifying these bacterial virulence properties. One attempt does that based on mechanisms of action. What makes it a bit difficult also to classify is the shared activities that they possess. But if one looks at the mechanisms of action, in order to mention them, then we have one group based on mechanisms of action. One factor can function in one or more ways, and they may be grouped as one group adherence factors. These are also referred to as adhesins. And as that name implies, they enable a bacterial cell to adhere to the cells of tissue surface in humans. So whenever one is exposed to these organisms, it's always a surface. It could be a surface inside the person, or it could be a surface on the skin. But whichever it is, the adherence factors or adhesins that enable bacterial cells to adhere to the cells of the human tissue on the surface. There are many, but the obvious ones are pili or fimbria, which I believe were covered when we did the structure of the bacterial cell. They are found in some bacteria and not in others. And these are regular pili or regular fimbria. There are some other types of pili, but these are the regular ones. These are the organelles of adhesion. So most of the time they are associated with gram-negative bacilli. Most of them possess these pili. And secondly, the surface of the organism, there are some binding molecules. This would be on the bacterial cell surface. And there are also some bacteria which have capsules. And on those capsules, there are also some components which are important when it comes to adhesion. Components of bacterial capsule and components of bacterial cell surface. Mostly surface and some of the surface antigens. Then another group, based on the way they act, this group is referred to as aggressive factors or aggressins. They enable the pathogenic organisms to overcome or evade host defense mechanisms. So they are aggressive towards the host tissues. and aggressive against the defense mechanisms that will try to eliminate them or stop them from spreading. So aggressins or aggressive factors include surface proteins and carbohydrates. Again, these are components of the surface covering of the bacterium. The capsule also functions as an aggressive factor in that it prevents the interaction where a phagocytic cell is supposed to recognize some components of the bacterium itself. The capsule forms a barrier and does not enable that phagocytic cell to recognize certain components of the cell surface, and that would be a step that is necessary before phagocytosis can take place. Then secondly, thirdly, there are some enzymes or enzyme-like substances. They act like enzymes. They might not be actually enzymes. So enzymes and enzyme-like substances are also aggressive factors. And types possessed differ among the bacteria, and they enable the organism to invade and multiply in tissues. This invasion and multiplication in tissues is what we refer to as invasiveness. So enzymes and enzyme-like substances increase invasiveness. Then there are some aggressive factors which act intracellularly. There are some bacteria which cause intracellular infections. They are associated with intracellular location. It could be in phagocytic cells like macrophages and others. So they survive and cause disease from that location. So they have some aggressins or aggressive factors which enable them to survive and multiply within phagocytic cells. And these aggressins which act intracellularly are limited to some intracellular pathogens. So we have mentioned the group of two groups, aggressins and aggressins. And the third group, what referred to as bacterial toxins. They are properties of some pathogenic bacteria, not all bacteria possess them. Some do, some don't. And then there are two types. They are classified into two types. One group are exotoxins, and the second group are endotoxins. This group, we start with the exotoxins. These are diffusible products released by some gram-positive bacteria or secreted, and the majority are secreted by gram-positive bacteria. So we started by saying not all bacteria possess these toxins. And then the exotoxins, some gram-positive, the exotoxins that we encounter, most of them are products of gram-positive bacteria, but not all gram-positive bacteria produce exotoxins. And then there are also a few gram-negative organisms which are capable of producing exotoxins. And they are produced by a bacterium. They are synthesized within the bacterial cell in the cytoplasm and secreted, so the action is outside that bacterial cell somewhere, the tissue target in that particular person. Then they are not uniform. Each type of bacteria, among the bacteria that produce exotoxins, each one produces its specific exotoxin. But there are some characteristics they share, some properties of exotoxins that are known. So they have some properties in common. One, they are proteins structurally. As proteins, Some of them are even enzymes or have enzymatic activities. So they are made of proteins structurally, and then as proteins, most of them are heat labile. Proteins are denatured by heat. So the majority are susceptible to heat and they are damaged by heat. So they're described as heat labile. That happens readily. And then there are a few which resist heat for some time or to a higher temperatures. And these few are described as being heat stable, but they are still susceptible to heat, but it's only that they are a little resistant. Then thirdly, these are products which are synthesized and liberated from the cytoplasm of live multiplying bacteria. That is one which is able to carry out some synthetic activity or synthesize materials within its cytoplasm and release them. So it has to be live, and more so when they are live and synthesizing, they are associated with multiplication. And then they are strongly antigenic. They form strong antigens. In other words, they stimulate production of antibodies more easily. And they can be treated in such a way that they lose their toxicity, but they retain their structure. And when it is treated, maybe by heat or chemicals, in such a way that it retains its structure, which looks like that of the actual toxin, but it's not toxic. It is called toxoid. So these exotoxins can be converted to toxoids. And these toxoids are useful in the medical field. They are used for immunization. And where there's a toxoid, when you'll be handling an organism that can produce a toxin that can be changed into a toxoid and can be used for prevention of the illness, it will be mentioned when you cover that specific problem. Then they are highly specific for certain tissues. They have target tissues on which they act. So we say they are released, maybe via the blood, they reach that target tissue, and that is where they act. And they are highly potent. One needs some very small quantities to see the effects. So they are high in potency. And then they are effectively neutralized by antitoxin. So that toxoid that can retain its structure, it can stimulate the immune system to produce antibodies. And those antibodies which are produced against the toxoid are effective against the actual toxin. So that is the antitoxin. And then the synthesis of the majority is coded for by extra chromosomal genes. That is the genes, most of them, not all of them. Some of them are actually coded for by the chromosomal genes. That is the main chromosomal material in the bacterium, or the nucleoid. And then most of them are coded for by genes which are not in that chromosomal part of the bacterium, extra chromosomal. Some of these are acquired, like the plasmid. It's important in coding for the production of some of these exotoxins. So not all of them again, some of them, most of them. There are also a few that are coded for by the chromosomal. So we said there are different substances specific to the organism that has released them. Activity and effects of bacterial exotoxins on tissues. The mechanisms of action and effects on tissues depend on the specific exotoxin. But there are many different ways through which they act, how they enter that particular tissue that they're going to cause damage in, But just to mention examples of how they might act, there are some which cause inhibition of protein synthesis in the tissue cells at the site of action, that is host tissues. The protein synthesis is inhibited, then they cannot survive. And then there are some exotoxins which interfere with the normal nervous response to stimuli. And then there are many exotoxins which act on the intestinal tract, and there are some with enzymatic activity. So when you cover the organisms which produce exotoxins, and some of them, you might hear the mechanisms through which they act. For others, you might have to look it up, but they all act in different ways. Then the endotoxins, these particular ones are contained in the cell wall of gram-negative organisms. So they are only associated with gram-negative bacteria. They form part of the lipopolysaccharide component, and they are liberated from the cell wall of dead or disintegrating gram-negative. So they also have some properties which distinguish them from the exotoxins. Being lipopolysaccharide, they are heat stable. They are not susceptible to heat, like the exotoxins, which are proteins. They cannot be converted to toxoids, and they don't have any specific tissue targets and receptors. Their toxicity is moderate and the potency is low, and they're poorly or non-antigenic. Even if one has the antibody to it, they're not easily or effectively neutralized. And being part of the bacterial cell component, the synthesis is directed by chromosomal genes. That gene that directs the synthesis of the cell wall. So the effects of the endotoxins. These generalized with variable manifestation. So where there's a light infection by gram-negative organisms and only a few of them disintegrate, one would have a small quantity of it in the blood. And that goes into the system that is associated with pyrogen release. So that would just trigger the release of pyrogens, leading to fever. But where there is a heavy infection, maybe something happens and most of those organisms disintegrate at the same time, one would have high concentration in the blood, and that interferes with the normal physiological mechanisms in multiple systems, leading to severe manifestations. So that concentration will cause abnormalities in immunological and other defense mechanisms, and also hematological processes. So the illness that results there is a very severe illness with multiple tissues or organs being involved, because the endotoxin is released into the blood circulatory system, and it operates from there, and it reaches many tissues, and they can be affected vastly by that concentration of the endotoxin. So hypotension and endotoxic shock are some of the manifestations or complications associated with high concentrations of endotoxins. So apart from those groups, 3 groups, there are also other bacterial properties which contribute to virulence they do not quite fit into any of those three, but it might not be direct effect on virulence, but they have some influence. They contribute to virulence. One such activity is the ability of that pathogenic bacterium to obtain iron. And this iron is obtained from the host that is infected or from the infected tissues. It is a characteristic of most pathogenic bacteria, and they obtain that iron through release of some small carrier proteins, molecules, which obtain iron from the infected tissues. And once the iron is obtained, it is utilized by the organism because it's essential for the nutritional requirements of that pathogenic organism. And when its nutritional requirements are met, it multiplies better and the illness becomes worse. So the availability of iron supports the organism's growth, and it also enables production of many extracellular products because the organism is operating at its level, a high level. It produces these products involved in virulence. Some of their production are influenced positively by the presence of iron, others are influenced negatively. But the iron, once it's available, it supports the growth of the organism and all the activities it carries out, including production of these extracellular products. Some of these extracellular products are exotoxins, which we have mentioned. So it has to operate maximally nutritional demands met, and then it will produce the exotoxins that are harmful to the individual. Another important property that may not be directly involved in virulence, but has an influence on virulence is plasmid. And what is important here are the genes that it contains, what they code for. One, there are some plasmid genes that code for resistance for certain antimicrobial agents, specific agents. So these genes are referred to as R factors. And an organism there could be an infection or an organism already establishing itself in tissues, and it has been detected and treatment started. And then it acquires some of these genes through the genetic material transfer. It acquires the R genes once the treatment has been started, and that antibiotic might be one that is affected or by the R genes or R factors. So there will be resistance, and this might not become obvious to the person giving the antibiotic. So treatment continues, but there is resistance that leads to continued growth and spread of infection despite that treatment. So it might have been susceptible initially, but it has become resistant and it might not be obvious, and then it continues to grow. Virulence, and then formation of sex pili and ability to transfer genetic material. This process is again made possible by the genes contained in plasmids. And the genes that are being transferred among them might have the alpha, so that will spread that resistance to other organisms. And thirdly, plasmids may influence virulence by what we have already mentioned, the production of some of the substances which it codes for, other characteristics which contribute to virulence, including exotoxin production. We said most of these are coded for by genes which are contained in the extra chromosomal part of the bacterium, and plasmid is one such part of the bacterium that contains genes that are significant, coding for exotoxin production. And thirdly, another property that is important when it comes to virulence is change of surface antigens. Some of the bacterial cells in a population of bacteria, where once it's an infectious process going on, there's a population of bacteria involved, and some of them, they multiply very fast and they become very many within a short time. And whenever you have a large population of bacteria in a certain part, some of them would undergo mutation. So some of the bacterial cells in a population of bacteria involved in an infectious process undergo mutation. And the mutation that is significant for our subject today is one which causes alteration of the genes coding for the antigenic structure. So in that large population, you get some organisms undergoing mutation. That mutation causes alteration of the antigen. So the antigenic component is important in this particular organism. So you get new strains of organism with altered or a different antigenic structure. So subsequently, the newly formed antigens possess what shall we treat? Yeah, the newly formed antigens possess different antigenic determinants from those of the previous cells, the original organisms that infected initially, that the human tissue recognized, the immune system recognized, then something new comes in that population. So you get new antigens which are not recognized by when the antibodies are released against the previous antigens, and therefore they're not eliminated. So the new strains with new antigens, they are not eliminated, they continue to multiply. And the illness or the infection becomes serious. So this change of surface antigens, all in all, it is a process which enables the bacterial cells with new antigenic determinants, which results from mutation, enables them to evade the immune defense mechanisms and to continue causing the infection. So we have covered pathogenicity, we have covered virulence. The third component of this lecture is infection. And this, again, we said there's an overlap in meaning or in usage. It can be used interchangeably. And infection is a process necessary for an organism to cause disease. We have talked about infectivity, which is related to infection. But sometimes this word is used to mean disease. And it is used a lot, and even by us, sometimes we use it when we mean disease. But strictly speaking, it is one, it involves sustained presence of an organism in a specific part in or on humans. This specific part can be on the surface, including the skin or mucous membranes, or it can be in the inner tissues. And then the sustained presence translates into establishment. It establishes itself in that site and begins to multiply and build up a population at that site, and that is the process referred to as colonization. And this is infection, with or without disease manifestation. So there are some factors related to the host with regard to infection. There's host and tissue specificity. There are some organisms which are known to be either restricted in the type of animals they infect. You'll be hearing of some bacteria which are strictly human pathogens, only associated with humans. You'll hear some which are animals. There are some which can come from animals to humans. But there are some which are obligate human parasites or pathogens. So there's the type of animals they infect can be specific, and also in humans, you can have specific tissues which are associated with them. Most of the time, primarily, whenever the infection starts, it starts in a specific tissue and it can develop into a severe disease in that tissue. Some of them remain restricted to those tissues, others can spread to other tissues, but they start from somewhere almost all the time. So these are host specificity and tissue specificity. So the reasons for these host parasites or tissue specificity are not clearly identified. So some might think of availability, some have thought of availability of oxygen or nutrients, but it is not quite established. While some organisms prefer certain tissues. So sources of infection and reservoir. So when we talk of a source of infection, we have a specific infectious process in mind, and you're thinking of the site from where that organism came from before it got into a specific host where it is causing the infectious process. So the source here might not be the very beginning. That one we don't know. But you talk about a source of infection when you have a specific infectious process going on. You want to know where that organism came from before it got into this person. And then reservoirs. So reservoirs, these are normal growth habitats of organisms, which can be a site on an animal, or it can be part of the environment. So when you have specific infections with clinical manifestations, we said infections might not have obvious manifestations, but where you have clinical infections in humans, these infections or infectious processes can be classified according to the source of the organism involved as exogenous or endogenous infection. So the exogenous infections, the organisms involved, are derived from the sources outside the infected person. And among these, it could be a person who is already ill from the effects of that organism, patients. It can be human carriers that we mentioned earlier. It can be an animal, or it can be the environment. These are exogenous sources, and the infections resulting from there are referred to as exogenous infections. The second group are endogenous infections, and for this, the organisms involved are derived from a site within the same individual. We had mentioned the complications of a symptomatic carrier state. One of them is disease process developing eventually from those organisms. So it can be pathogenic organisms in an asymptomatic carrier causing disease in that same carrier. And secondly, there are some organisms that you're going to cover in the next lecture, which are potential pathogens. Particularly, you're going to cover the bacteria that are referred to as normal flora. So they some of them are potential pathogens and are capable of causing disease in the same individual that is harboring them. So when you look at infections in general, there are some modes of transmission that are known. These are important modes of transmission in case one needs to intervene or stop the spread. Sometimes there's an outbreak or protect others around the other. You need to know how those infectious agents are spread. So the modes of transmission and spread of pathogenic bacteria are determined largely by the site infected and the infected organism. So here we are talking about the majority. There are few exceptions, but for the majority, if you look at the respiratory tract infections by bacteria, majority of causative bacteria are carried in infected secretions. And this could be from the upper part, the throat, could be from the lower part, the lungs, but eventually there are secretions that reach outside from those sites. And this can be released through sneezing, through coughing, through speaking, and they come out in droplets, a spray of secretion. So this can reach the susceptible person either directly or via contaminated items. This could be a surface or clothing, any contaminated item. So this again is the majority. That means there's a small minority that don't follow this pattern, but most of them could occur this way. Then if you look at infections involving the skin and wounds, these are spread through contact with the infected part or material from that part or via contaminated hands or other items. So some of these are common sense. We're just mentioning them for completion's sake. Then there are genital tract infections. Okay, genital tract is part of the human body. It can be infected by many different organisms. Most of the ones, the majority of infections involving this part, which are encountered, are mostly due to bacteria, which are transmitted during sexual contact. So they are seen in adults. The transmission occurs through mucosal surface to mucosal surface contact or through secretions. Then gastrointestinal tract infections, the majority of bacteria involved are acquired through ingestion. Some of the bacteria are passed out in feces. It could be diarrheal, stool, but depending on the practices in that particular population, how they handle the waste products, there can be contamination of water by these organisms that are passed out. They are initially ingested by someone, passed out by the feces, get water sources contaminated, the water contaminated, also hands can be contaminated, food or other items. And from these contaminated areas, they can be re-ingested, and the process repeats itself. And that is what is referred to as fecal oral transmission. Then there are some bacteria which involve bacterial infections, which involve the blood circulatory system. Not very many of them, but there are some that have been identified. Among them, there's some illnesses due to bacteria which are found in the blood due to arthropods. When they feed, they carry the organism from one person because they feed on blood, and when they feed on the next person, they release the organism. So that type is referred to as arthropod-borne or vector-borne infections. So these are spread by blood-sucking arthropods during feeding. And secondly, there are some infections in the bloodstream of expectant females. Some bacteria from time to time are encountered, associated with the blood circulatory system of a female, and she might be expectant. And this particular female is capable of being a source of an organism that is transmitted transplacentally, or horizontal transmission to the expected baby. And thirdly, there's a possibility of organisms in the blood spreading via donated blood. This was significant in the early years before methods were developed which can screen donated blood for various organisms. So it is documented as a possibility, but in this day and age, all donated blood is screened for various infections. Among some of them are bacterial. So if it is not screened, the possibility of transmitting a bacterial pathogen contained in it from the donor to a recipient are higher than if it was screened. So transmitted, you'll then encounter the one or two, mainly one bacterial agent, which could be in the blood, and it's screened. The blood is tested for before it is accepted after donation. And the last part is laboratory acquired accidental infections. These are possible because in the laboratory, bacteriology laboratory, one is handling a specimen, is a diagnostic laboratories. There, one is dealing with the specimen and analyzing it for bacterial infection. One doesn't quite know which bacterium is in it, and one can easily get exposed to it. But in the diagnostic laboratory, they have safety precautions, which are documented and people are taken through before they start handling these specimens. But also in the laboratory, the learning processes in like your practical sessions, what used to happen used to have organisms grown artificially on artificial media. And those organisms are alive and capable of causing infection. Again, there are safety measures which are adhered to lower the chances as much as possible of those handling that situation from being exposed to those organisms. So another potential source in the laboratory are laboratory animals. There are some people who have, as part of the laboratory, they have a section where they handle animals. These animals are used to test many things, organisms, drugs. They are handled by many different people. And being animals also, a person could be exposed to infection from them. But again, there are safety measures which have been put in place that people adhere to. So the chances of laboratory acquired accidental infections are reduced. But people are made aware, should there be an accident, then one knows what to do. So you can still hear me? How come after this projection was transferred, I'm not getting those messages I was getting? Is it related to your devices? You can hear me? You can't. Are we still together? Yeah. Where are the charts? Julie, the answer is yes. Oh, after transferring. At ease. If I send this to it is not going. So Maureen, in the making of the toxoid, it's not necessarily chemical. That is one way, but it would be used for immunization if you're listening to the lecture. It would be used. Yeah, is Maureen listening? Maureen. where we can see the bond. The bundle. Zoom. When sharing.